Hello everyone and welcome to this high value masterclass. My name is Aditya and I'm a redox biologist by training. In the series of high value masterclasses, this is fourth masterclass where I'll be describing you about beta oxidation of fatty acids and their energetics. This class also highlights one of our recent research articles that has been accepted for publication on development of a tool and an integrated formula for bioenergetics. So let's start. We all know that the beta oxidation process is a catabolic pathway where fatty acids particularly get cleaved at the carbon number 3 or beta carbon sequentially, and they eventually turn into water and carbon dioxide, generating a large amount of energy. So in this whole lecture we'll be first looking at introduction, then the overview of the process, then four-step cycle, the core of beta oxidation, metabolic fate of reducing equivalents which are generated during this process, energetics, and unusual oxidation processes like alpha oxidation, omega oxidation, and peroxisomal beta oxidation. And as I had told you, one of the recent article, a student-centric method for calculation of fatty acid energetics, integrated formula, and web tool development will also be highlighted here. So friends, let's take a deep dip into oils and fats. Some of you might believe that fats are really very dangerous for health. And that's not wrong to believe that, because dysregulation of fatty acids and their metabolism may lead to serious health concerns. But that also doesn't mean that you should be scared of fats and fatty acids and they're not at all beneficial for us. Indeed, some of the very important life processes occur because of presence of fats and fatty acids. We will realize that our most of body organs, such as heart, muscles, they operate only because of the presence of fatty acids and their oxidation. So they are not as bad as they appear. Now first let us look at from where do we get fats and fatty acids. So before talking about that, let me describe you what is a fatty acid. A fatty acid is a molecule that contains carboxyl group COOH that is connected to a long chain of alkyl groups CH2, 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 maybe like 10 or 12 or even more. So by chemical definition, we call fatty acids as long chain carboxylic acids. Now there are two categories. One category is saturated, another category is unsaturated. How do we define saturated and unsaturated? Primarily on the basis of the presence of a double bond or its absence. So if there is no double bond, they are called saturated fatty acids because every carbon atom on them is fully saturated by hydrogen and they're unable to carry out any addition reactions. However, they can undergo substitution reactions. On the other hand, unsaturated fatty acids, they have at least one double bond, mostly in cis orientation, that gives rise to this kind of kink shape in the fatty acid. And if there are more, they will be called as polyunsaturated fatty acids. So we can obtain these fatty acids from two places. Either we can get them from dietary sources, from food that we eat, and in the dietary sources, again, we can have either saturated fatty acids or saturated fats. For example, in butter, we have myristic acid. From coconut, we can get palmitic acid that has got 16 carbon atoms. Stearic acid, which is mostly obtained from animal fat that has got 18 carbon atoms in them. And arachidic acid, which is from plant origin, that has got 20 carbon atoms in them. Besides saturated fatty acids, which are mostly obtained from animal fats or animal sources, except for the coconut oil, which is also saturated, majority of the plant-based oils or vegetable oils, they contain a rich amount of unsaturated fat. So now let's look into the examples of some of the unsaturated fatty acids, which are mostly of plant origin. The first most important one is a palmitoleic acid that has got 16 carbon atoms in it and one double bond at ninth position. So it's represented as delta 9. But if you count it from the other side, from the omega carbon side, this double bond will be at seventh position and you call it as omega 7 fatty acid. Besides this, you have oleic acid, which has 18 carbon atoms and one double bond at ninth position, so it's omega 9. And both these, as they have only one double bond, they are called as monounsaturated fatty acids. But there are some fatty acids which have more than one double bond. They are called PUFA or polyunsaturated fatty acids. One such example is linoleic acid, which has two double bonds and 18 carbon atoms. These double bonds are at position 9 and 12, making it omega 6 fatty acid. Then alpha linolenic acid, which has three double bonds in it and having 18 carbon atoms. These double bonds are at position 9, 12, and 15 respectively, making it an omega 3 fatty acid. Then we have arachidonic acid, which is an unsaturated version of arachidic acid. This has got four double bonds at 5, 8, 11, and 14 position, and this has got to be called as omega 6 fatty acids. So these are most common type of fatty acids that we obtain from the dietary sources, whether it is a vegetable diet or it is a non-vegetarian diet. But if you talk about the another way by which we can obtain fatty acids is the endogenous synthesis. So humans and other mammals are capable of synthesizing fatty acids endogenously in their liver. And mostly humans and other mammals, they can only synthesize saturated fatty acids because they do not have ability to introduce double bonds or unsaturation. So very little or no unsaturated fatty acids are endogenously synthesized in higher animals and mammals. However, bacteria and plants, they have the systems to introduce unsaturation, and they can of course synthesize all these kind of unsaturated fatty acids that we talked about. So this is where we get fatty acids from. And this is our routine, like 30 to 40 grams of fats is a daily routine in the normal diet. American diets or fast food eating people, they can have more fat and fatty acids in their diet, but this is how we obtain all of them. Now, one important question arises. Are fatty acids or fats better than carbohydrates, especially in terms of the energy efficiency? The answer will be yes. Fats are more efficient. You can look at the calorific values, you will realize that carbohydrates per gram can generate only 4 kilocalories of energy, whereas fats, they can generate almost twice the amount of the energy generated by carbohydrates, which is about 9 kilocalories per gram. So why does that happen? Why fats can generate more energy per gram? Because of two primary reasons. First, they are highly reduced. If you look at the carbohydrate structure, you will find that carbohydrates have got a lot of oxygen in them in the form of hydroxyl groups. That means they are oxidized, partially oxidized. So if they are already oxidized, the further oxidation will generate lesser amount of energy. Whereas if you look at the fats, they have got long chain of alkyl groups, which is completely reduced, and therefore oxidation of those reduced molecules or reduced groups can generate considerably more amount of energy. The second important factor for volume to energy ratio is their hydrophobic nature. We all know that humans and other organisms have aqueous systems, and most of the molecules are suspended in aqueous environment or water-based environment. So fats being highly hydrophobic, they cluster together and they remain away from the water, clumped together in highly compact form. So this compactness of fats and fatty acids make them generating more energy per unit volume as compared to the carbohydrates. Now there is one more advantage of why fatty acids and fats are better than carbohydrates in producing energy, because besides producing energy, they also produce more metabolic water that we will see how organisms utilize during their metabolic requirements. So that's yet another advantage why fats have been chosen by the nature as more efficient and much better energy fuels than carbohydrates. So let's now look at the power of beta oxidation. How does it fuel life, and what different life forms depend upon beta oxidation, especially in some of the most extraordinary features of the life? You might have heard about a bird called golden plover. This bird is capable of traveling as much as 3,300 kilometers in one go, that's around 35 hours, at an average speed of nearly 60 miles per hour, where it has nearly 250,000 wing beats. And believe me, all the energy comes from beta oxidation of fatty acids. Similarly, another bird called Arctic tern, which is a migratory bird and travels almost one pole to another, that also solely depends upon beta oxidation. And then golden-necked hummingbird of America, which is also a migratory bird, it can have a non-stop flight from Central America to Mexico in one go. So all these exemplary, exceptional features and beauty of life is fueled by fatty acids. So this reveals the power of beta oxidation in the living world. Now, if you further look at some of the other organisms living in extreme conditions like wild Mongolian jabril, which doesn't ever drink water because it doesn't require water, as all the metabolic water can be produced from beta oxidation of fatty acids, that has a remarkable lifestyle, all owing to beta oxidation. Killer whales, despite the fact that they live in sea, they generate most of their water from metabolism of fatty acids. Yet another organism that you know and you are familiar with, camel, ship of the desert. It has a large reserve of fat in its hump, and all the metabolic water, or most of the metabolic water in the time of emergency, is generated by beta oxidation. So it can survive for a long time without consuming water. So that's yet another example of the power of beta oxidation in nature. Now, if you talk about humans, us, then most of our life and some of the very important organs of our body also rely on the power of beta oxidation. Almost every cell that contains mitochondria is virtually capable of utilizing fatty acids as fuel, provided that it can internalize those circulating fatty acids. Most importantly, our heart. You must wonder that people say fats and fatty acids are dangerous for heart. Indeed, they are dangerous. Only dysregulation is dangerous. But if you look at the biochemistry of beating of heart muscles, you would find that as much as 90% of the energy of cardiac muscle contraction comes from fatty acids. However, it is possible that depending on your requirement, daily routine, and dietary habits, the heart muscles can quickly switch from fatty acid beta oxidation to glucose metabolism, and this demand can reduce from 90% to 60% in some cases. Similarly, resting muscles and our muscles during the mild exercise, they also mainly depend upon fatty acid oxidation. However, as I told you, certain cells which do not have mitochondria, like erythrocytes and islets, they cannot utilize fatty acids for beta oxidation. Then some of the organs and tissues which are not permeable to fatty acids, such as brain, which has a very tight blood-brain barrier, is not capable of permeating those fatty acids into it, therefore cannot utilize those fatty acids. However, a derivative of fatty acid metabolism called ketone bodies are often used by brain during the glucose deprivation conditions. So, with this, I would say that we have enormous examples throughout the life. Our own survival, the very important organ, heart, which pumps almost day and night, 24/7 to keep us living, is mostly relying and being fueled by fatty acids. So, this is the importance of understanding the beta oxidation of fatty acids. So now let's talk about historical aspect of beta oxidation, how the name beta oxidation came and what it actually means. So this is almost a century old pathway. And you should know this person and you should thank this person, Franz Noop, because of him this pathway was known in as early as 1905. He lived from 1875 to 1946, and there is a very good connection between beta oxidation and dog because the pathway of beta oxidation was actually deciphered by using biochemistry of dog. Now, what Franz Noop did, he actually utilized odd chain fatty acid rich diet, which was labeled with phenyl groups, and even chain fatty acid rich diet, which was also labeled with phenyl group. You should know that this is the structure of phenyl group, and this is used for quantification. Now, after doing this, the dogs urine was collected. So it might seem funny to you, isn't it? But believe me, this was one of the first biochemical labeling method where the chemical labeling and tracing was used to look at the biochemical fate. And hats off to this guy who made it possible in those days when nobody thought about labels. So the outcome of this result was then evaluated, and the urine of dog was studied. It was found that when dog was given odd chain fatty acids, this resulted in production of hippuric acid, whereas if the dog was given even chain fatty acids, that resulted in the formation of phenylacetic acid. Now, how does it answer the question that how beta oxidation takes place and why should we call it as beta oxidation? So if you look at the chemical breakdown, you can observe here that there is a long chain of fatty acids which will be broken down from one end, and in the end, the carbon between alpha and beta should be broken in order to give rise to a compound called benzoate, which then combines with glycine in order to be detected, that forms hippuric acid. Similarly, if you look at the breakdown of even carbon numbered fatty acids, here it is alpha and beta carbon atom where the breakdown will take place, and this will give rise to a compound phenylacetate, which when combines with glycine during detection forms phenylacetic acid. So had it not been two carbon breakdown, you would have never obtained phenylacetate. So in results, obtaining these phenylacetic acid and hippuric acid by Franz Noop made it confirmed that whenever fatty acids are breaking down, they are doing so in the set of two carbon atoms. And since it is happening between alpha and beta carbon atom, and during the process beta carbon is getting oxidized, the process was named as beta oxidation. We will learn in more details eventually in coming slides that how exactly beta oxidation takes place and how this beta carbon is getting oxidized during this process. So this was the historical background. Although in 1905, when he did this experiment, most of his hypothesis could not be validated until 1950s. So it was the year 1950 when most of the work done by Franz Noop was confirmed, and then it was made sure that oxidation of fatty acids mostly take place between alpha carbon and beta carbon, that is carbon number 2 and carbon number 3 from the carboxyl side. So this is how the name beta oxidation was given to it. Now let's look at the overview of whole pathway. How does it happen? And then we will look in sequence one by one each of the steps. So you know that fatty acids are prevailing everywhere. They are present in our body. They can either be taken from dietary sources or they can be generated endogenously. But the site of beta oxidation is inside the cell. So those circulating fatty acids must actually first enter into the target cell. That makes the first step. Then if you look at the thermodynamics, which we will understand later in coming slides, we will find that these fatty acids need to be energetically activated. So this activation of fatty acids is the next step during the oxidation of fatty acids. Now, the process of oxidation or beta oxidation is coupled to energy generation in electron transport chain, and it is destined to occur in the powerhouse of the cell, that is mitochondria. So mitochondria, which is the primary site, apart from that, it also occurs in peroxisomes that we will discuss at the later point of time. So the third step would actually be the entry of these activated fatty acids into mitochondria. That is a detailed process which requires a carrier molecule that we will discuss in detail. And now, once these activated fatty acids reach inside the mitochondria, they undergo a sequential oxidation process at beta carbon. Now, this process is a four-step process which generates acetyl coenzyme A after each cycle, and two reducing equivalents NADH and FADH are also reduced. Now, this acetyl coenzyme A then enter into TCA cycle, which is Krebs cycle, and During this cycle, they are completely broken down into carbon dioxide, generating more reducing equivalents. Now, all these reducing equivalents, NADH, FADH, all these go into the electron transport chain, pump the protons into the transmembrane space, and then eventually resulting in production of large amount of ATPs, which powers the cell. So this is the overview of oxidation, which we are going to learn in coming 20 to 30 minutes. And we will talk about each of these steps individually, one by one. So here is the first step of beta oxidation, that is entry of fatty acids into target cells. Now what we have learned so far, that fatty acids may come into our body either through exogenous mechanisms, that is through dietary route, or they may be synthesized directly in the liver. But the site of their activity, site of their usage is in a variety of tissues and variety of cells. We learned by now that they can be utilized in your skeletal muscles, in heart, cardiocytes. So now transport is an important aspect. These fatty acids must be transported from the organs like intestine to liver to different parts of the body. And we know that the solubility of lipids is quite less. It is somewhere in the micromolar range. But these lipids are made soluble in bloodstream by conjugating them with proteins, and that results in the formation of complex known as lipoproteins. Well, that's the topic of another discussion where we can discuss in detail about the transport. That's quite an independent topic to be discussed in detail. But right now, I'll just curtail myself here with 2 points, that most of the exogenous dietary fatty acids and fats, they are packaged in the intestinal enterocytes in the form of chylomicrons, and those packaged chylomicrons then get transported to different target tissues. Similarly, the fatty acids which are synthesized in the liver, they also get packaged in the form of lipoproteins called VLDL, which are then transported to the target organs. Now, near the target organs, there are capillaries, blood vessels, which have ability to break down or rupture these lipoproteins and then allow release of free fatty acids just near the cells. So now it is important that how these things enter into cell. Yet another thing, some of the fatty acids may be coming from your stored reserves in the adipose tissues. So there also most of the fatty acids, they get conjugated to a protein called albumin that makes half of the blood proteins, and that carry those lipoproteins, making them more soluble somewhere in the millimolar range, from micromolar range to millimolar range. So these soluble molecules then reach the target cell and they now have to enter into these cells. Now, although we know that plasma membranes are hydrophobic in nature, and any hydrophobic molecule can cross the diffusion layer and that can move across the concentration gradient, but we require that transport to happen much more effectively and much faster. We need fatty acids to enter in larger amounts. So, human cells or cells of other animals also have specialized carriers or transporter proteins which are present in the plasma membranes. There are three important classes of carriers or transporter proteins which are found in the cells. One of them is known as fatty acid transporter or FATPs, which mainly belong to SLC27A class of genes. And there are many different subclasses, as many as 25 genes are involved in this process, and variety of proteins encode for different SLC27A. They express in tissue specific or cell specific manner, and different type of FATPs can be present in different cells. So this transports much more quickly these fatty acids into the cell. Another kind of transporter is known as long chain fatty acid acyl coenzyme synthesis, which also performs passive transport into the cells. And the third category of transporters is known as fatty acid translocases, or simply FATs. And in other terms, they are also known by the name CD36. So they also cause a transport. Now, once these fatty acids have entered into the cell, they are now ready for the process or initiation of beta oxidation process. But still, there's one hurdle. Let's look into that. Now, the hurdle is thermodynamic aspect. If you look at various biochemical pathways, whether it is glycolysis, TCA cycle, or any other metabolic pathway, you would find that generally a first step is the activation step where the whole process has to be made thermodynamically favorable. You might be aware about the concept of Gibbs free energy, where the Gibbs free energy of whole metabolic pathway should be negative. That means the process should be exergonic in order to carry out that spontaneously. So these fatty acids which have just entered the cell, they are not activated and they do not have a good amount of free energy in them, and therefore they cannot drive the process of oxidation by themselves thermodynamically. Now, in order to make them active, these fatty acids generally combine with a high energy molecule known as coenzyme A, which is synthesized endogenously in the body. This coenzyme A can couple to these fatty acids, resulting in the formation of a molecule called acyl coenzyme A. But this is an energy requiring process where ATP is converted into AMP, and that's equivalent to the loss of two ATPs that we will look in energetics portion. So let's have a quick look at mechanism of how this process happens. You can see here the structure of adenosine triphosphate or ATP that initially combines with the carboxyl part of a fatty acid. Now, as you can see here, this oxygen, which is highly electronegative, this attacks the first phosphate or alpha phosphate of ATP, shifting the bonds of this PO onto the oxygen and resulting in eventual cleavage of pyrophosphate. So this pyrophosphate goes away from here, and this AMP then got conjugated with fatty acid. So this is known as fatty acyl adenylate, which is an intermediate during the activation of fatty acids. Now the byproduct is pyrophosphate. This whole reaction is catalyzed by an enzyme called fatty acyl coenzyme A synthetase. That's primarily a ligase enzyme, and this first part is called as adenylation reaction. So adenylation eventually makes this process activated for next set of reaction where this phosphate can be additional AMP can be removed, and then coenzyme A, which is another high energy molecule, can get conjugated to it. So in the next step, this coenzyme A, which is also a high energy molecule that attacks this positively charged carbonyl carbon or carboxy carbon, thereby shifting these electrons of the single bond to the oxygen. And during this process, coenzyme A gets connected or gets conjugated to the fatty acid, resulting in the formation of our activated fatty acid molecule called fatty acyl coenzyme A. And as a byproduct, you have AMP subsequently released. So you have ATP turning into AMP in this process, pyrophosphate is released as byproduct, and fatty acid, which was unactivated or a low energy molecule, got converted into a very high energy molecule called fatty acyl coenzyme A. Now, as I told you, why this process is happening? Because we have to make this whole process spontaneous. Now, the second part is known as thioester formation, which is activating this molecule. And if you look at thermodynamics of this, we would find that as a very important characteristic of most of the biochemical reactions, they are coupled. That means one exergonic reaction is coupled to an endergonic reaction. So what we see here, that delta G of acyl coenzyme A formation alone is nearly plus 31.5 kilojoules per mole. That means the process was non-spontaneous otherwise. But the hydrolysis of ATP to AMP, that contributed as much as 32.3 kilojoules per mole, which is an exergonic process, making the overall process with delta G value of 0.8 kilojoule per mole in negative. That means it is spontaneous process now. But this is quite a little amount of exergonic energy that cannot drive the complete pathway. So the remainder of energy comes from hydrolysis, further hydrolysis of pyrophosphate, because this pyrophosphate can also, with the help of inorganic phosphatases, can turn into 2 moles of phosphates, and that has got delta G of minus 33.4. So this adds to the free Gibbs energy of this whole process and makes this whole process highly exergonic and makes this fatty acyl coenzyme a very active molecule to be converted into subsequent reactions by the downstream pathway. So this was the second important part of beta oxidation, that is activation of fatty acids. Now we should remember that this process is still taking place in the cytoplasm. But you remember that oxidation process should happen in mitochondria, which is powerhouse of the cell, because most of the reducing equivalents that will be generated, they will be present in mitochondria, and they can be converted into ATPs, or they can generate energy only in mitochondria. So it is essential to transport this activated fatty acid or acyl coenzyme A into the mitochondria. Now that's a tough part. Why tough part? Because if you look at the mitochondrial membrane, that is made up of two components, that is in inner mitochondrial membrane and outer mitochondrial membrane, and in between that there is a hydrophilic region which is called intermembrane space. So let's try to look at this whole process of transport of acyl coenzyme into mitochondria with an analogy of crossing a river. So suppose you have a river and somebody want to cross this river. It is much better that if we have a boat around there. But if you leave that boat unattended, somebody might just destroy it or steal it, or maybe the person who is trying to cross the river is not able to board on the river without any assistance. So let's say there are two guards which are placed on either side of the river who are maintaining the boarding and embarking or disembarking of the people on either side. One person is helping passengers to board on this boat, and another person is helping people to get down from this boat. So this will make the life of people easier to cross the river. And people can also swim across the river, but those people who cannot swim the river, those who are afraid of water, they will of course not be able to cross it. So lipids are molecules which are afraid of water. They cannot swim across the hydrophilic region, so they need a boat. And that is the analogy which explains how fatty acids can be transported into mitochondria. Now this boat is a molecule called carnitine. You can see here the black part in this image, this tetramethyl hydroxy butyric acid. This is carnitine. And this carnitine can get conjugated with the fatty acid. So fatty acid is the passenger and this is a boat which will be carrying it across the intermembrane space. So let's look at the process. This is the outer membrane of the mitochondria, which is more or less permeable to most of the molecules. It has a higher permeability. Then inner mitochondrial membrane is relatively less permeable, and in between that there is a hydrophilic region called intermembrane space. So as I told you, we have a boat called carnitine and we have a passenger called acyl coenzyme A. But now we need someone to allow acyl coenzyme A to board the boat. We need assistance. So that assistance is provided by an enzyme called carnitine acyl transferase, which is analogous to this guard which is placed over here. And what it does, it actually combines or connects these two things. There is another counterpart or another analogy with the guard, carnitine acyl transferase 2 on the metric side, which will help in disembarking the people from the boat or removing the acyl coenzyme A or fatty acid from the carnitine. So, in a process catalyzed by carnitine acyl transferase, and if it is a palmitic acid based fatty acid, it's also known as palmitoyl acyl transferase. Now, during this process, this acyl coenzyme part is conjugated to carnitine. As you can see here, carnitine was having a hydroxyl group at this carbon, and this hydroxyl group get connected to the carboxyl group of fatty acid, and the coenzyme A part is released back in the cytoplasm. So as you saw here, in this process, there is also a requirement of translocase through which this carnitine can move. And as you can again see this boat went inside. Carnitine took the acyl coenzyme A, acyl group or the fatty acid carnitine, which is also called as fatty acyl carnitine, and the coenzyme A part was left outside in the cytoplasm. Now we have a separate pool of coenzyme A inside the mitochondrial matrix, which can again combine with this fatty acid on reaching the mitochondrial matrix, and that thing will be catalyzed by another isoform of carnitine acyl transferase, which is on the inner mitochondrial membrane side called CAT2. Now this conjugated and this made it free. So this is the disembarkment or release of acyl coenzyme A into the matrix, and now carnitine, this boat is free, but this boat has to be made available back into the other shore of the river. So this boat or carnitine then goes back through that same translocase. It comes back into the outer part, or you can say into the cytoplasmic part, and then it is ready to carry out the new fatty acid into the mitochondrial matrix. So this was the process how fatty acids are transported into the mitochondrial matrix. Now once these fatty acids have reached the mitochondrial matrix, which are activated, they are now ready to undergo the 4-step biochemical pathway, which we actually call as beta oxidation. Now when we have already learned about the basic process of transport of these activated fatty acid into the mitochondria, now let's talk about the core 4 steps or the core part of the beta oxidation. As I told you that this beta oxidation is a cyclic process where each cycle results in breakdown of 2-carbon compound that's called acetyl coenzyme A from activated acyl coenzyme A. Now in this process, the first metabolite that we have seen is acyl coenzyme A that has just entered into the mitochondria. Remember, this process is taking place inside the mitochondria. We'll talk about the enzymes and how these complexes are attached to mitochondrial membrane in the coming few minutes. So this acyl coenzyme A, as you can see here, this is the coenzyme A part attached here, and this carbon is known as carbon alpha. The red colored highlighted carbon is known as carbon beta. So during this whole cycle, oxidation is mostly going to take place on this carbon, which gives this process name beta oxidation. Now, these carbon can also be numbered according to new IUPAC nomenclature. This one will be 1, this one will be 2, and this one will be 3. So you can call beta carbon as delta 3 carbon as well. The first reaction of this pathway is a reduction step, which is catalyzed by enzymatic activity called acyl coenzyme A dehydrogenase or ACAD. During this process, 2 hydrogens will be removed from alpha and beta carbon successively. So removal of the hydrogen atoms from this result in a new molecule called trans delta 2 enoyl coenzyme A. As you can see on the second carbon, there is now emergence of a double bond, which will be useful to understand when we'll talk about unsaturated fatty acids. So already we are creating an unsaturation at alpha beta carbon or carbon 2 and carbon 3. Now, as this process is a reduction process, protons are being released. So, reducing equivalents like FAD, they take up these protons and become FADH2, which become high energy molecules and aid in generation of ATP. In the subsequent step, now it's the turn to add water to it. So that's a hydration step which is catalyzed by delta 2 enoyl coenzyme A hydratase activity. It is also known as crotonase. And there is an addition of water molecule HOH at alpha beta carbon. So as you can see here, the proton has added here on alpha carbon, whereas OH has added on beta carbon. So this is the first place where oxygen has added. So one step of oxidation took place here where hydrogen was removed. Then second oxidation took place here where oxygen is added on beta carbon, and that resulted in the formation of this molecule called L-beta hydroxyacyl coenzyme A. Remember that it is important to know here that this form of unsaturated compound is trans, which is quite unusual. Most of the double bond or unsaturated fatty acids are cis in nature, whereas this form is L form. It is a type of configuration of the molecule. Most of the molecules are in D form, but some molecules in biology are also found in L form. So L-beta hydroxyacyl coenzyme A is the second step or the second metabolite that is formed during this pathway. Then the third step is again a reduction step. You can call it as oxidation step. Now again oxidation will take place at beta carbon atom. So this time there will be again removal of two hydrogen atoms by enzymatic activity beta hydroxyacyl coenzyme A reductase. And this, as you can see here, the hydrogen has been removed. Now it's only double bond O left over here. And this compound is known as beta ketoacyl coenzyme A. We have to remember that R represents a long chain where there will be several CH2 depending on the number of carbon atoms present in the fatty acid. Now last step, during this reduction step, as you have seen, there is a removal of hydrogen atom. So these hydrogen atoms are then again taken up by reducing equivalents like NAD plus, and that turns into NADH. You may perhaps ask me that why FADH here and why NADH here, why not vice versa? So it depends upon the energetics and redox potentials of these molecules and also the cofactors associated with specific enzymes. So this place we generated one FADH2, and at this place we generated NADH as reducing equivalents, which will help in ATP generation through electron transport chain. Now the final step is very important because at this step you have the lysis of this molecule at beta carbon. So between alpha and beta carbon atom, now the lysis will take place, and that will result in the formation of two carbon compound called acetyl coenzyme A. And that step is of course known as thiolysis because it is aided with the help of a thiol intermediate and catalyzed by an enzyme thiolase or beta ketothiolase. So this is the acetyl coenzyme A molecule, which you are familiar with already when you studied about tricarboxylic acid cycle or Krebs cycle, and it's a substrate for TCA cycle. So we have to remember one thing, that during this process, another coenzyme A that interacts at this carbon, beta carbon, and gets connected with it. So imagine if COA or coenzyme A is getting connected at this place. Now this will turn the remaining part very much similar to this original compound called acyl coenzyme A. So in this step you will again be generating an acyl coenzyme A, which will be 2 carbons shorter than the original one. So suppose you started with 16 carbon atoms at this place. At the end of this complete cycle, you will be having a 14 carbon acyl coenzyme A. And acetyl coenzyme A, a 2 carbon compound, will be produced additionally. Now looking more detailed mechanism of the breakdown of this molecule or thiolysis, we can observe that the enzyme thiolase actually has got a thiol group or SH group that initially attacks this carbon. So this is your acetyl group, carbon 1, carbon 2, carbon 3. So this one is alpha carbon and carbon 3 is beta carbon. So basically beta carbon is being attacked by this thiol group. Now once it is attacked, the electrons of double bond oxygen move on to oxygen, making it negative, as you can see here. And this carbon, which becomes positive, becomes a center for nucleophilic attack by this enzyme. Now residues in enzyme cysteine that contains S will attack this carbon and will form this complex. Now this complex will then release this acetyl part from here by delocalization of electrons of oxygen and then delocalization of these electrons onto acetyl group, leading to the formation of free acetyl coenzyme A, which is released from the enzyme complex, and this enzyme still remain bound with remaining part of fatty acid that makes it enzyme thioester intermediate. Finally, this thiol intermediate is attacked by another coenzyme A, high energy molecule, and that coenzyme A get attached to this acyl group, or you can say remaining fatty acid, and finally it is again released as free acyl coenzyme A, making enzyme free. So you have seen in this step that how acyl coenzyme A was regenerated after losing 2 carbon molecule acetyl coenzyme A from here. So the number of carbon atoms got reduced by 2 in this process. This is the basic beta oxidation process. You can remember the steps by a short acronym like RHRT or OHOT, that is reduction, hydration, reduction, and thiolysis, or oxidation, hydration, oxidation, and thiolysis. That makes, I think, your life easier to remember what beta oxidation is. Let's now talk about how these enzymes are organized and where exactly inside the mitochondria this process is taking place. So as we have seen this complete process of beta oxidation in four-step reaction cyclic pathway, and we have four intermediates or four enzymatic activities. The first one was a dehydrogenase activity, second one was a hydratase activity, and third one was again dehydrogenase activity, and the last one was thiolysis activity. So in humans and other mammals, there are not four enzymes for this. Rather, we just have two enzymes. One that performs the first reaction, dehydrogenase reaction, and remaining three reactions are conducted by an enzyme that has trifunctional activity. So all the three remaining enzyme activities are present in a single complex enzyme complex, which is also known as trifunctional protein or mitochondrial trifunctional protein MTP, that has three activities of hydratase, enoyl coenzyme hydratase, and it has beta ketoacyl dehydrogenase activity and ketothiolase activity. This MTP is a heterooctamer, which is composed of 4 alpha and 4 beta subunits. Now, if you look at the organization of these things in the mitochondrial membrane, so all the enzymes of beta oxidation are attached to the inner mitochondrial membrane. As you can see here, the acyl coenzyme dehydrogenase is quite a big complex which is attached to inner mitochondrial membrane. And then if you talk about the trifunctional enzyme, which has been solved a number of times through X-ray diffraction, and recently in 2019 this structure has also been solved by Jung P. Kim et al. That appeared in PNAS in 2019. So I'm showing the same structure over here, which is an octameric unit. So you have alpha 1 here, alpha 2 here, and beta 1 and beta 2 here. So that's one unit, and such kind of two units are clubbed together. So you have 2 alpha 1, 2 alpha 2, 2 beta 1, and 2 beta 2 units here. And as you can see here, the green part, which is making this first half of the green part, which is making alpha 1, and the remaining, this orange part, is making alpha 2. So you can see diagonally here that the green part, alpha 1, that actually has this enoyl coenzyme hydratase activity, and the diagonally represented alpha 2 units, they are representing HAD or hydroxyl coenzyme dehydrogenase activity. Now, the third activity of enzyme, that is thylalysis activity, is contributed by this beta subunit, and beta subunit is the one that helps in anchoring this enzyme into the mitochondrial membrane. So, in most of the organisms, beta oxidation is carried out mostly by two different set of enzymes, acyl coenzyme dehydrogenase and trifunctional mitochondrial protein, which has three enzymatic activities. Hope that makes you understand and realize that how this process is taking place inside the mitochondria. You can have a good glimpse of this three-dimensional view of protein, which is again presented by the author Jung P. Kim et al. Now let's talk about the fate of acetyl coenzyme A. So you are already familiar about TCA cycle or citric acid cycle, which carries out set of 8 reactions in which it successively break down this acetyl coenzyme A into carbon dioxide. As you can see here that acetyl coenzyme A will undergo a first reaction through oxaloacetate. It will make citrate and then it goes to isocitrate, makes alpha ketoglutarate, and during this step it's a reduction step where NADH is formed and carbon dioxide is also released. Then in subsequent step, you have formation of succinyl coenzyme A. That is again a reduction step where NADH is formed, and succinyl coenzyme A then turns into succinate where it generates 1 GTP that can also be turned into ATP further. Now this succinate then forms fumarate by another set of dehydrogenation reaction generating FADH2 reducing equivalent. Then fumarate takes up a water molecule, turning into malate, and then this malate turns into oxaloacetate, generating yet another NADH. So you can see in this process that these particular reactions, like succinyl coenzyme A to succinate, and then from succinate to fumarate, fumarate to malate, malate to oxaloacetate, these reactions are pretty much similar to some of the reactions of beta oxidation. As you can see here, first reduction happened, that first the oxidation happened, that means dehydrogenation happened, and then addition of water, and then again dehydrogenation. So you can recall the reaction number 2, 3, and 4 of beta oxidation, where you saw similar kind of pattern, reduction dehydrogenation process that was followed by hydration, and that was further followed by thylalysis. So these are analogous to reactions of beta oxidation. Now finally, let us conclude the overall integration of all those things, how these all reducing equivalents are fed into energy generation pathway, and how these two things, TCA cycle and beta oxidation, are clubbed together. They are actually happening together at the same place in the mitochondria. So as we have learned in the previous section that these enzymes of fatty acid oxidation are attached to the inner mitochondrial membrane. So the first enzyme, acyl coenzyme dehydrogenase, and the trifunctional mitochondrial protein, both of them, they generate or break down the long chain fatty acids into acetyl coenzyme A, generating some reducing equivalents like NADH and FADH respectively. Now, besides that, you have plenty of enzymes which are conducting this particular cycle of citric acid cycle or Krebs cycle. Now, during this process also, you have utilization of acetyl coenzyme A, and that acetyl coenzyme A is generating 3 NADH and 1 FADH, as you can see here, as well as 1 ATP, which is in the form of GTP here. Now all these reducing equivalents are eventually fed into electron transport chain, which is a subject of another discussion. Electron transport chain has got 4 complexes: enzyme complex 1, complex 2, 3, and 4. Succinate dehydrogenase, which is a part of citric acid cycle. It is the only membrane bound enzyme which is part of electron transport chain as well, and remaining enzymes are present in the mitochondrial matrix. So during this process, when the electron flows from one part of the electron transport chain to another part, some of the protons are actually pumped into transmembrane space. That's 4 protons from complex 1, 4 protons from complex 3, and 2 protons from complex 4. However, complex 2 doesn't pump any protons. Now these protons generate an electrochemical gradient, and this electrochemical gradient then drives the rotation or conformational change of this enzyme called ATP synthase. And when these protons return back, they generate high energy molecule ATP that powers the cell. So this was a quick glimpse to understand that how TCA cycle and beta oxidation go hand in hand. So if you're generating a lot of acetyl coenzyme A, they are actually working as a substrate for this TCA cycle and the ATP generation, both from direct reducing equivalents obtained from the cycle as well as from the TCA cycle. They all are fed into ETC and they generate ATP in this case. It's now time to look at the overview or energetics of beta oxidation, that how many ATPs we could generate during this complete process of beta oxidation depending on the number of carbon atoms in the fatty acids. So let us first talk about a simple case of those fatty acids which have even numbers of carbon atom in them. Let's say this is a long chain fatty acid, and you can see here that are some 16 carbon atoms over here. This whole acyl coenzyme A, or this activated fatty acid, will undergo a process of beta oxidation. And during the process of beta oxidation, as we have already seen, a cyclic events will take place and they will result in the generation of FADH2 and NADH in each cycle. And this acetyl coenzyme A, which is produced here, that will be fed into citric acid cycle or TCA cycle. And this will then lead to the generation of few more NADH, GTP, and FADH. So we can simply look at this overall process, and by using this simple illustration, we can try to generate a formula or calculate the number of ATPs in beta oxidation. So let's talk about each beta cycle. You can see here that during each beta cycle, a fatty acid is breaking down into acetyl coenzyme A. So let's say there were 16 carbon atoms here, and you required how many cycles in that case to split it up? Just check out. This is released in first cycle, in second cycle, in third, in fourth, fifth, sixth, seventh. So in seventh cycle, you will have the 2 acetyl coenzyme A generated in the end. So you can have intuitively a simple mathematical term for this, that if there are n number of carbon atoms or c number of carbon atoms, then the number of cycles required would be c by 2 minus 1. That means if you have 16 carbon atoms, like if you are talking about palmitic acid, in that case, you will require 16 by 2 minus 1. That is 8 minus 1, or 7 cycles. So I simplified this to 0.5 C minus 1, and equal to the number of cycles will be number of FADH2 generated and NADH generated in each of these beta oxidation cycles. So there will be 0.5 C minus 1 FADH2, 0.5 C minus 1 NADH2. And now let's talk about the fate of these acetyl coenzyme A. So as we can see here, that there will be 0.5 C, or half the number of carbon atoms, acetyl coenzyme A. So if there were 16 carbon atoms, there will be 8 acetyl coenzyme A that will be generated here. And if you look at the fate of TCA cycle, you can check here that each TCA cycle is generating 3 NADH, which is equivalent to 7.5 ATPs, 1 FADH, and 1 GTP. So now, if we want to generalize, or if we want to create a general formula based on this, what we can do is we can integrate all these things and do some mathematical transformations to get a common formula for finding out the number of ATPs by breakdown of even numbered carbon fatty acids. So let's see here that how we derive this integrated formula for saturated even number of fatty acids. So number of cycles were 0.5 C minus 1. Acetyl coenzyme A generated were 0.5 C. You multiply that with 10 because each acetyl coenzyme A is generating 10 ATPs per cycle. And then you have 0.5 minus 1 FADH generated from each cycle of beta oxidation. You multiply that with 1.5 as it gives you 1.5 ATPs. NADH gives 2.5 ATPs and there were 0.5 C minus 1 NADH. So you multiply this here, you get three equations. Now these are the total number of ATPs which you get after complete breakdown of even numbered fatty acid. You add these equations and do some simple maths, you get net ATP is equal to 7C minus 4. You have to remember one important thing here, that during the activation process there were 2 ATPs consumed. Actually, that was one ATP turning into one AMP, which was considered to be equal to consumption of 2 ATPs. So in order to calculate the overall ATP calculations, we have to subtract 2 from this formula, and therefore we can have net ATP from saturated even number fatty acids as 7C minus 4 minus 2. That turns out to be 7C minus 6. So this is something we postulated in this paper, and we can have a very general formula that can be used for calculation of ATPs for beta oxidation of fatty acids with even numbers. So if you have palmitic acid that has got 16 carbon atoms, you can simply multiply 16 with 7 and subtract 6 from it. You get 106. Similarly, you can do that for arachidic acid that has got 20 carbon atoms. 20 into 7 minus 6, that means 140 minus 6, that gives you 134. You can do that for 18 carbon atom fatty acids. 18 into 7 minus 6, and so on. So this is a universal formula that can be used for calculation, direct calculation of ATPs much more quickly and efficiently. Now the next important thing is, right now we talked only about the fatty acids that contained even number of carbon atoms. Usually humans and mammals synthesize even numbered fatty acids, but some of the plants and marine organisms, they also synthesize fatty acids which are having odd number of carbon atoms. So what do you think? A fatty acid with 16 carbon atom would be better, or it will yield more energy, or a fatty acid with 17 carbon atom would yield more energy? Please answer this. 16 will yield more energy or 17C will yield more energy? How do you think? What will be the answer? Now that's an important question, need to be answered here, and at the end of the discussion we'll be able to answer this question. So let's talk about another fatty acid which is one carbon longer than the previous one. As you can see here, in the previous one you had 16 carbon atoms, and here it is representing 17 carbon atoms. So let's check about the fate of this. This will also undergo similar kind of beta oxidation process. That means it will have a routine cycle. In every cycle you will be generating one acetyl coenzyme A, and you will be producing NADH, and the acetyl coenzyme A which are broken down will eventually go into TCA cycle, and when they will go in TCA cycle, as you know, they will be generating 3 NADH, 1 GTP and 1 FADH. So now the question is, how many cycles would happen if there are 17 carbon atom? Anyone please answer? Will there be 7 cycles or 8 cycles? Let's look into this. So in the first cycle, this acetyl coenzyme A gone. Second cycle, another. Third cycle, yet another. Fourth one. Fifth one. Sixth cycle. Seventh cycle. Now do you think 8th cycle will take place or not? This is now a 3 carbon compound left. Will there be 8th cycle or not? The answer is no. Reason: this 3-carbon compound or propionyl coenzyme A, which is left in the end, is too small to act as a substrate for the last enzyme you remember, thiolase. So it cannot be recognized by it, and therefore it is left unturned or uncut, and it is therefore an end product. So what is the impact of this on ATP generation in this whole process? Let's talk about this. So what will be the number of cycles? We can have this general formula intuitively. The number of cycles will be C minus 1 divided by 2 minus 1. That means the number of cycles in 17 carbon fatty acid will be same as that of there were in the previous 16 carbon atom fatty acid. So it's C minus 1 divided by 2. That was the number of cycles in case of the previous one. Now we have to subtract minus 1 to get the same number of cycles. So 17 minus 1, that is 16 divided by 2. That gives you 8. 8 minus 1 gives you 7. So we will have 7 cycles for a 17 carbon fatty acid. Now this can be mathematically simplified as 0.5 C minus 1.5. So depending on what is the number of cycles, each beta cycle generates one FADH and one NADH. So these many cycles will generate these many FADH and these many NADH per cycle. Similarly, there will be C minus 1.5 acetyl coenzyme A as well. 0.5 C minus 1.5 acetyl coenzyme A. So those acetyl coenzyme A will also generate the 10 ATPs per cycle. Now what is left is this molecule. What will happen to this? Will it also generate some energy? Yes, of course. There is a metabolic way to turn this into energy. Propionyl coenzyme A, which is a 3-carbon compound, is actually converted into a 4-carbon compound, succinyl coenzyme A. And this succinyl coenzyme A can be then fed into TCA cycle to generate some ATPs. But how many ATPs? Let's look into this. Usually each succinyl coenzyme A will produce 1 GTP, 1 FADH2 and 1 NADH. But if you look at the detailed mechanism of formation of the succinyl coenzyme A, you would find that there is consumption of 1 ATP at the activation step. So this propionyl coenzyme A, when it combines with CO2 with the help of enzyme propionyl coenzyme A carboxylase, that hydrolyzes 1 ATP into ADP and turns it into methyl malonyl coenzyme A, which is the S configuration, and that need to be converted into R configuration. Just you can see here flipping of alkyl group and carboxyl group, changing the configuration, and this then turns into or rearranges into another isomer called succinyl coenzyme A. So during this whole process, you saw an utilization of 1 ATP. So if you calculate the yield of ATP coming from this propionyl coenzyme A, you will find that there is 1 NADH, 2.5 ATP, 1 FADH, 1 ATP, 1 ZTP, which is equivalent to 1 ATP, and 1 ATP is consumed. So in total, you have 4 ATPs which are coming from this remainder part of fatty acid. But we have to remember that the number of acetyl coenzyme A in this case are 1 less than the number of acetyl coenzyme A which were produced in the previous example. The mathematical transformation of this whole energetics, and let's try to get one integrated formula which can be used for calculating ATP yield. So now what we have seen already, that number of cycles in case of odd numbered fatty acids would be 0.5 C minus 1.5, and the number of acetyl coenzyme A will be generated equal to the number of cycles. So as we know that each acetyl coenzyme A when undergoes a TCA cycle can generate 10 ATPs, so you simply multiply it with 10 to get the total number of ATPs generated from acetyl coenzyme A. Further, FADH would generate 0.5 C minus 1.5 into 1.5 ATPs, because per FADH2 there is 1.5 ATP generation from ETC, and NADH will generate 0.5 C minus 1.5, that's number of cycles, into 2.5, these many ATPs. Now, in addition to this, there is also a palmitoyl coenzyme A, which will be generating a gross ATP of 5, but during the carboxylation step, as we have seen previously, 1 ATP was being consumed. So net ATP production will be 4 from this molecule. Now, if you simply add and do some mathematical transformation, equation 1, 2, 3, and 4 get added, and we achieve net ATP yield as 7C minus 17, I'm sorry. So the previous rule that we applied to the even numbered fatty acids also applies here, because even the odd numbered fatty acids will get activated to acyl coenzyme A by using 2 ATPs or ATP turning into AMP, and therefore the net formula or overall integrated formula would be 7C minus 19. So on one hand, where we have seen the integrated formula for even numbered fatty acids as 7C minus 6, here for odd numbered fatty acids it is 7C minus 19. So now let's talk about the comparison of two. Which is better? Which is more efficient? Whether odd numbered fatty acids generate more energy or even numbered generate more energy. So you have seen the two pathways, you have seen the overall integrated formula. For even numbered, it is 7C minus 6. For odd numbered, it is 7C minus 19. Here that which one would generate more energy and which one would generate less energy. So if you look at a classical example or a most prevalent example of a saturated fatty acid, palmitic acid, which has 16 carbon atoms in it, and that is mostly obtained from the palm oil or coconut oil, you can apply this formula here, 7C minus 6, and you get 7 into 16, that's number of carbon atoms, minus 6 gives you 106 ATPs. But if you look at successive fatty acid, which is having 17 carbon atom, that's margaric acid or heptadecanoic acid, which is mostly obtained from margarine, it's a substitute for butter, the ATP yield in this case will be 7 into 17, number of carbon atoms, minus 19, that gives you 100. And that's a good reason why nature chose even numbered fatty acids for most of the higher organisms for generation of ATPs. Despite the fact that C17 fatty acids had more number of carbon atoms, but they did not have more ATP yields. So in most cases, you will see that higher organisms which synthesize their own fuels, which synthesize their own fatty acids, they rather than preferring odd number fatty acids, they prefer even number fatty acids. So that was all about saturated fatty acids which don't contain any double bond. Now, is it the same case with unsaturated fatty acids? Do we also oxidize them? And if we do oxidize them, what are the challenges that are posed? And what is the difference in metabolic breakdown or pathways? And what are the new enzymes that are needed for those pathways? That we will look shortly. So here is something that we are going to discuss now about unsaturated fatty acids oxidation. Now, before we start discussing about oxidation of unsaturated fatty acids, let me just take you back to this oxidation cycle of fatty acids. You must have to recall here that step number 2, where dehydrogenation was taking place from the acetyl coenzyme A to delta 2 enoyl coenzyme A, you already witnessed that a compound with double bond was being created, which is named as delta 2 enoyl coenzyme A. So this is carbon number 2 where double bond is present, or we can say that it is between alpha and beta, which at later point of time will be cleaved from here after 3 more subsequent reactions. So first there will be hydroxylation or addition of water over here, and then again there will be dehydrogenation, and then there will be cleavage. So if there are unsaturated fatty acids already in the nature, and if they have some double bond at some point of their molecule, some place, then they can directly become a substrate for this second reaction. I mean, they can skip the first reaction and they can directly enter into the second reaction, isn't it? That's quite simple. But that doesn't happen in reality. Can anyone tell me the reason why double bond containing fatty acids cannot directly enter at the second level? There are two reasons. The first reason is, as you can see here, this is a trans form, whereas most of the double bonds that are found in fatty acids are in cis configuration. So first challenge that is achieved or that is observed is presence of a cis bond but not a trans bond that refrains those double bond molecules or unsaturated fatty acids from directly entering into the beta oxidation. Now the second challenge is position of the double bond. So you can see here that the double bond should be present at the even numbered carbon, that means between 2 and 3, or you can call it as alpha and beta. What if the double bond is present at this place between 3 and 4? So when you have long chain fatty acids, you might have seen that there are 16 carbon atoms, 18 carbon atoms, and in unsaturated fatty acids, the double bond may be present either at ninth position, 11th position, 12th position, 15th position. So as far as it is at the second position, that will be in the ambit of alpha and beta, because when you will break in the pieces of 2, that even numbered double bond will come exactly at alpha and beta. But if it is at odd numbered position, it will actually come between beta and gamma when you will break that down into small pieces. So we will have a glimpse of it. We will just look at it. As you can see here, this is representing a straight chain fatty acid. Now, this is having 16 carbon atoms represented here by small dots. And you can see here that there is a double bond. Now, this double bond is between 9 and 10. Let's take for example, because most of the unsaturated monounsaturated fatty acid like palmitoleic acid or oleic acid, they are delta 9 and they have double bond at ninth position. So that was the reason I considered it at 9 and 10. Now this is placed at odd number. So obviously if you break them off, it's not going to come at alpha beta position, but it will come at gamma beta gamma position. So let's see what happens during the beta oxidation process. Now this delta 9 double bond which is present here, this is one specific case, and these fatty acids will be called as odd numbered, or you can say double bond placed at odd numbers. But other than that, there can be some fatty acids which can have double bond placed at even number, as you can see here between 12 and 13. This is delta 12 bond. So there can be two categories of fatty acids, or there can be two categories of double bond that need to be handled in unsaturated fatty acids. One at odd numbered positions and other at even numbered positions. And then there can be more complex scenarios when there are polyunsaturated fatty acid. So there can be some double bonds which will be placed at odd number, some double bonds which will be placed at even number. And each of these will be handled according to the procedure which we'll be discussing now. So let's first talk about the odd numbered double bonds. I mean, a double bond placed at odd numbered carbon atom. So what will happen in this whole molecule, how the process will start? You already know now that this is, let us say, carbon to which coenzyme A is attached. So this is your alpha carbon atom and this is your beta carbon atom. So during the first reaction, a double bond will be introduced between the first and second carbon, or alpha and beta carbon. Now what is the next step? Hydratase reaction, OH will be added here, and then after that there will be dehydrogenation reaction. This double bond will be lost, and then there will be eventual release of this acetyl coenzyme A. Now you saw that this whole process was taking between alpha and beta, or carbon number 2 and 3. Now subsequently, the same procedure will follow in the next cycle. You will again introduce a double bond here. This double bond will then will be between alpha and beta and will release acetyl coenzyme A. In third cycle, again you will have the same thing. But now tell me whether fourth cycle will happen or not. I need answers from you. Yes, anyone please? Fourth cycle will happen right now or not? It seems that it can happen, right? You can place a double bond here and you can then cleave it because it's clear. But in reality, when there is a double bond at this place, and during the reaction, when you place a double bond between alpha and beta, the hydratase enzyme cannot further place a hydroxyl group over here. So this is not the correct substrate for hydratase. So dehydrogenation might occur, but hydration reaction will not happen. So this is the step where the problem will arise. And in order to solve this, in order to prevent the stoppage of the reaction or to continue this beta oxidation process, it is important that this double bond should be somehow removed. Now the solution is quite straightforward. Instead of entering the beta cycle, this whole thing, even the first reaction doesn't happen here, but if you switch or replace this double bond from this position, that is beta gamma position or 3-4 position, to 2-3 position somehow, that is alpha beta position. So what would happen? You just replace this double bond from gamma delta beta gamma position to alpha beta, and then you made this molecule a substrate for the second reaction, as I was mentioning in the beginning. Now during this process, what also happens is the conversion of cis bond to trans bond. So prior to this, it was beta gamma cis bond, but now this has turned into alpha beta trans bond. So now after this is converted into alpha beta trans bond, this is now a substrate for the second enzyme, hydratase enzyme, and that can easily be cleaved off again. Now let's have a look at the detailed mechanism. How does it happen? So you can see here, this is a gamma beta bond, and during the fourth cycle, when the problem was happening, an enzyme particularly known as enoyl coenzyme isomerase, which is required in addition to the 4 enzymatic activities which are required in beta oxidation. So this enoyl coenzyme isomerase is an additional enzyme which will perform this function of converting this beta gamma bond into trans alpha beta bond or delta 2 bond. So delta 3 to delta 3 bond, or you can say 3-2 enoyl coenzyme isomerase performs this function. Now we have to remember one important thing, that this substrate which was released and this reaction which produced 1-acetyl coenzyme A, during this reaction the remaining substrate or remaining molecule will not go into the first step. Rather, it will directly go into the second step of the cycle, because there was already a double bond that was created. So you will start the process from delta 2 enoyl coenzyme A instead of acyl coenzyme A, and therefore 1 FAD that was being generated will be reduced. So in that case, when you are doing enoyl coenzyme isomerase activity, in that case, you will not be having this first dehydrogenase activity, and therefore 1 NADH, 1 FADH will be produced less in number. That counts during the energetics process. Now after this, rest of the process is same. Remaining acetyl coenzyme A will be released sequentially after each beta oxidation. So depending on how many such kind of double bonds do you have in the process, if you have many such double bonds which are present at odd numbered, each of these will be processed in the similar fashion. So although we are talking about monounsaturated fatty acids with one double bond example, but this can be extended to polyunsaturated fatty acids where there will be a double bond that is placed at odd position. So are we clear? Double bond placed at odd position can be shifted to even position and cis can be turned into trans by simple isomerization reaction, and then it can become directly the substrate for the second reaction of beta oxidation. Now, little more difficulty appears when you have this kind of situation when double bond is placed at even numbered position. So, as you can see here in the first reaction, that's like usual. Second reaction is also usual. Third cycle is also usual. You produce acetyl coenzyme. Then fourth cycle also is usual. But now, do you think fifth cycle will happen properly? It seems that it can, but actually not. Because if you place a double bond here, still at this position there is a double bond. So that still creates a problem, although it is at a distant place, but that's almost like 2 to 3 carbon away, and that does not allow this acetyl coenzyme to break down. Now, what is the solution for this? You can see here, if you break this, this is at the right place, alpha beta position. But despite that, you cannot introduce a double bond here and you cannot create a hydration reaction at this place. So what is the solution? Your enoyl coenzyme isomerase won't work here, and even if it work, it won't place this double bond at the right place. So there is no use of this enzyme now. We need another enzyme. What we need to do? Think about it. So we can eliminate this double bond from here. And this 4,5 double bond is eliminated by using a specific set of enzyme that is called as reductase enzyme. So you reduce this place, that means introduce hydrogen, and this double bond will be gone. And once this double bond is gone, now it is okay for you to carry out this process. Let's look this in detail how this happens. So you have this usual 2,3 double bond, that is beta double bond, which is introduced during the regular process, and this was 4,5 double bond was previously present in the molecule. Now, there are two different ways in which it can happen. In the bacterial cells, there is a single enzyme that can directly convert this in one reaction into this molecule. It can reduce this. And you should know that during reduction process, it consumes NADPH and that turns into NADP plus, another reducing equivalent. So that is generally considered equivalent to the loss of around 3.5 ATPs. Whereas in the mammals, this process happens in two-step reaction. First, there will be 2,4-dienoyl coenzyme reductase that will reduce this bond, but unfortunately, reduction of this bond will lead to the creation of this bond which is at 3,4 position, as you can see here. So this 2,3 bond now switched to 3,4 bond when reduction happened, but you again require this double bond to be at 2,3 position. So that is performed by 3,2-enoyl coenzyme isomerase. So you have two enzymes that are needed to complete this process in mammals. And then, as you can see here, this is your delta 2-enoyl coenzyme that's represented here, and that can be directly fed into beta oxidation cycle, which can undergo routine process. So unlike the previous procedure where you had double bond at odd position and you did not require any energy, in this case, when you have a double bond at even position, you require energy. That means you require reducing equivalents NADPH, which is almost like equivalent to the loss of 3.5 ATPs. Another difference is in the first case, here the substrate that was generated was directly fed into the second step. But here you will not have the second step entry directly because the first unsaturation was already created by acyl coenzyme A dehydrogenase, and therefore the loss of one NADH, FADH that we were considering in the previous part, we will not consider that part. So let me just quickly look at this again. In the first part, where you had double bond at odd position, isomerase can flip this double bond from beta gamma to alpha beta position, and that makes it a substrate for delta 2 enoyl coenzyme A, therefore eliminating the need of first step of beta oxidation. So you are at the loss of one FADH2 during this process. Whereas if you have a double bond at even numbered carbon atom, in that case, just in a previous cycle or penultimate cycle, you will have to remove the double bond by using a reductase enzyme, and in case of mammals you use combination of 2,4-dienoyl reductase and 3,2-enoyl coenzyme A isomerase, which directly gives you the the delta 2 enoyl coenzyme A. But in this case, you have only a loss of around 3.5 ATPs in the form of consumption of NADPH. But FADH is still generated just like the conventional way. So this is how beta oxidation of unsaturated fatty acids takes place. Now let's get to the formula. And the remaining acetyl coenzyme A will just break in the conventional manner that I just wanted to show to you. So depending on how many double bonds you have at even position, those many times this process will happen. Now let's look at a mathematical transformation of this whole calculations for MUFA. So that's quite simple. We already have seen in the previous mathematical transformation that ATP calculated for even numbered fatty acid was 7C minus 6. For odd numbered, it is 7C minus 19. So what we only need to do here is that in each cycle, suppose we are talking about MUFA, where in most cases the double bond is present at odd number. If you look at the examples, you will find that most of the monounsaturated fatty acids are delta 9. So we are talking about that situation where double bond is present at odd position. So in that situation, you just have a loss of one FADH per cycle. That means whatever is the number of double bonds at odd position, you will have the loss of one FADH that is equal to the loss of 1.5 ATP. So we can simply have this factor 1.5D that can be subtracted from this old formula to get a new formula for ATP calculation of unsaturated fatty acids. And interestingly, this integrated version can also be applied to saturated fatty acids, because if you place the value of D as 0, there is no double bond, then it is automatically reduced to 7C minus 6. So that was the interesting thing we deduced. We got a common formula for monounsaturated fatty acids, or even it will work for PUFA if all the double bonds are present at odd numbered position. And simply by putting the value of D as 0, you can deduce the formula for saturated fatty acid. Same is true for odd numbered carbon atoms. So we know already the logic. There is a palmitoyl coenzyme A which is being produced. So that leads to this formula 7C minus 19, which we have already seen. And then again, if you subtract 1.5D from it, you get this formula 7C minus 19 minus 1.5D for calculation of ATP for odd numbered fatty acids which have double bond, all the double bonds at odd positions. So let's now look at the case of polyunsaturated fatty acids. Now, what do you mean by polyunsaturated fatty acids? That means they have degree of unsaturation at multiple places, so they have multiple double bonds. Some of the common examples that you know from the nature are these kind of molecules. One is this, which has got 18 carbon atoms in it and it has got double bonds at two positions. And another one again has 18 carbon atoms but has got three double bonds here. So you have this molecule called as linoleic acid, which has got 18:2 or 18 carbon, two double bonds, one at ninth position, other one as 12th position. And if you count it from the other side, it's also called as omega 6 fatty acid. If you look at the another one, that's alpha linolenic acid, which has got 18 carbon atoms but 3 double bonds, and these bonds are present at position 9, 12, and 15 respectively. This is called as omega 3 fatty acid. So this is one of the essential fatty acids that we need in our diet, and particularly the pregnant women require a large amount of ALA or alpha linolenic acid because we cannot synthesize it. Now, how do these fatty acids undergo breakdown? So we have to remember the strategy that we talked a minute ago, where we saw that how these double bonds are treated and dealt with. So we have two ways of dealing with these double bonds. So we have two ways of dealing with this double bond. One is a way to look at odd numbered double bond. Another way is to deal with even numbered double bond that we have seen just a minute ago. So let's take an example of a fatty acid that has got both the kind of double bonds. As we can see here, linoleic acid has got both kind of double bonds. One is at odd number and other one is at even number. Now let's see what happens if this is a conventional fatty acid which has got 18 carbon atoms, and you see here that one double bond is present at ninth position, other one is present at 12th position. So let's talk about the case what happens. These are the two double bonds. Now the process of beta oxidation would begin here. This part of first carbon would be connected with coenzyme A to get activated, and then during the first step a double bond would be created between alpha and beta carbon atom. We can also call it as 2,3 carbon. Now, just after the double bond has been created, sequence of events would occur. There are three reactions that you already know by now, and then this would be splitted off as acetyl coenzyme A. So that completes cycle number 1. Now cycle number 2 will be identical and follow in the same manner. You will again have acetyl coenzyme A being released. Then cycle number 3, it will also happen in the same manner, and you will release acetyl coenzyme A. But the problem will appear in cycle number 4. Now, why would you have a problem? Because if you create a double bond here, there will be two double bonds, alpha beta and beta gamma, that will make this molecule inaccessible for delta 2 enoyl hydratase. And therefore, this problem has to be resolved. So this problem can be resolved, as we know from previous discussion. There is an enzyme called enoyl coenzyme A isomerase that can shift this bond from beta gamma position to alpha beta position. And once this bond is shifted here, it's now a substrate for delta 2 enoyl coenzyme hydratase, and then it can easily split up and can result in the formation of acetyl coenzyme A. What about the next cycle, cycle number 5? So it seems that cycle number 5 should be fine because this is alpha beta carbon, and here one double bond can be introduced and this can go away. But in practice, it's not true. A double bond introduced here would have a difficulty, and the enzyme which utilizes this particular substrate, that is hydratase enzyme, would find it difficult to carry out the reaction if there is another double bond at the subsequent position. So it is important to first remove this double bond from here in order to carry out this step, and that problem is handled by another enzyme which is called dienoyl coenzyme reductase. So this dienoyl coenzyme reductase removes this double bond by performing a reduction reaction or adding two hydrogen atoms, and the carbons get saturated, so there is no requirement of a double bond now, and that's done by this enzyme at an expense of NADPH, which is usually considered to be equivalent to ATP of 3.5 ATPs. Now, once this problem has been resolved at the cycle number 5, then the problem is over, and this can break off as acetyl coenzyme A, and remaining carbon atoms can then undergo 3 subsequent beta oxidation cycles and can also be released as acetyl coenzyme A. So this was the process, and now we saw that how both strategies that we looked previously were involved here in breakdown of polyunsaturated fatty acids. So depending on how many odd numbered double bonds you have, how many even numbered double bond you have, these strategies can be repeatedly used. If you have all the double bonds that are at even number, only the strategy 1 would be used. If you have all the double bonds present at even numbered, strategy 2 would be used. And if you have one odd, one even, one odd, one even, so accordingly strategy 1 and strategy 2 could be used. So if you talk about the second one, that's alpha linolenic acid, it has got one bond at 9, another bond at 12, another bond at 15. So bond number 9 and bond number 15 would be dealt by strategy 1, that's enoyl coenzyme isomerase, and bond number 12 will be dealt by strategy number 2, that's dienoyl coenzyme reductase. So this is how polyunsaturated fatty acids undergo beta oxidation. Now, if we talk about the energetics, then we know that this whole process is going on in the cyclic manner, and we have deduced a common formula based on the number of cycles. So how many cycles do we have here? If we have even number of carbon atoms in a fatty acid, the formula was you halve the number of carbon atoms and subtract 1 from it. So 18 divided by 2 minus 1, that gives you 9 minus 1, 8 cycles. And we see here that we have sequentially done 8 cycles that resulted in production of 9 acetyl coenzyme A. So the formula comes out to be 0.5 C minus 1 cycles and 0.5 C acetyl coenzyme A. Now these acetyl coenzyme A would be fed into TCA cycle in mitochondria and would generate 10 ATPs, as you already know now. But there is one situation here, that is cycle number 4, which was carried earlier. There we did not had a complete beta oxidation, but we had a partial beta oxidation. The first step did not happen. So we are at the loss of 1 FADH at this step when we are using strategy 1. And when we are using strategy 2, that at the cycle number 5, we are actually at the expense of 1 NADPH, and this expense is concurrent with not losing an FADH. So when you are utilizing NADPH, that cycle is not partial, but it's a complete cycle. So you can say that you're not losing FADH here despite of having a double bond, but you are losing NADPH. So net loss would be 3.5 loss and 1.5 gain. So it will be loss of 2 ATPs. I hope I made myself clear at this place. So now we can say collectively, if we want to do a mathematical transformation or we want to have a formula for this, we can say that you will use a strategy 7C minus 6, that's for saturated fatty acids, minus 1.5D, that accounts for the loss of 1 FADH. So this gives you number of ATPs depending on how many double bonds you have. And if you have double bonds at even positions, then you're going to do 7C minus 6 minus 2D. But if you want to include both these things, if both kind of bonds are present here, so you can do this as 7C minus 6 minus 1.5D at odd number minus 2D at even number. So this would DE would represent double bonds at even number, and this would represent double bond at odd number. And we have done some simple conversions and simple conventions based on our experience, and based on commonly used fatty acids in the biological system, we have reduced this formula to 7C minus 6 minus 1.5D minus 2 within brackets D minus 2. So this applies to a limited number of fatty acids which are present in biological systems. However, you can make use of this formula for more general and universal purpose. So now this was all about energetics. Let's come to what we have hypothesized. We have come up with an integrated formula. We have reduced all the calculations and simplified everything and came up with this particular formula. A general formula, net ATP calculation for even numbered fatty acids, you can make use of 7C minus 6 minus 1.5D minus 2D minus 2. So this first part represents the formula for saturated, because if you place D as 0, this whole term will become 0 and you will reduce it to 7C minus 6. And if you have unsaturated fatty acids, particularly all the bonds are of same nature, either at odd position or at even position, then you can use this factor. For odd positions, which are commonly known factors, you can use minus 1.5D. For even positions, you can use minus 2D. And then if you have PUFA, which is again based on the conventional, commonly occurring fatty acids, you have to add this factor minus 2D minus 2. And for odd numbered fatty acids, those which have odd number of carbon atoms in them, we just have one difference, that we have 19 in place of 6, and then accordingly we can calculate the ATP. So we also developed a calculator code by using HTML, and then we got a web tool for it. And it's now simple. You can calculate all those things by simply placing those values into that calculator. Moreover, this is based on the observed type of fatty acids, but if you want to make a more universal formula, so this is the more universal formula, 7C minus 6 minus 1.5DO, where O represents the odd number double bonds and here E represents the even number double bonds. And then you can replace 6 by 19 to get this formula for odd numbered carbon atoms. Now this is the highlight of the calculator we have developed. The link is already provided to you in the comment box. You can anytime use that link, and you can make use of that calculator to calculate the number of ATPs. You can simply add number of carbon atoms and number of double bonds and click on submit, and you get the number of ATPs. This totally works on the formula that we have developed, and we are glad to share that our entry has also appeared in the Wikipedia for citing the energy yield. After learning how this beta oxidation process goes and looking at the energetics, let's quickly look at how this beta oxidation process is regulated inside the cell. What are the control centers, and how at genetic level and metabolic level this pathway is controlled, how to make a check. So we know that this pathway is taking place in different compartments. We have involvement of cytoplasm, we have involvement of mitochondria, and fatty acids which undergo oxidation, they actually come from outside the cell through transporters. So we have multiple things where we can make a control. If you think intuitively, you will recognize that we can make a control at this point. We can increase the import from these transporters, we can express them in large number, or we can improve the movement of fatty acids, activation of fatty acids and their movement into the mitochondria, or we can increase the enzymatic activities inside the mitochondria itself, which are controlling this beta oxidation pathway. So these are the common tricks by which the beta oxidation could be controlled. And there will be, of course, molecules like carnitine, fatty acyl translocase, which will be involved in this process. So let's see through this process. You know that the carnitine acyl transferase 1 and 2, they are involved in the transport process, and you have the process of beta oxidation that is going on inside the mitochondria, which is successively happening. So the first genetic control comes from the genes which are present in the nucleus. Now, among various regulators, one of the most important regulator is AMPK, that is AMP activated protein kinase, which is not only the controller of this pathway, but it also controls several other pathways. Now, what it can do is it can activate PGC1 alpha, which is a kind of peroxisome proliferator activator receptor co-activator. And besides AMPK activation, this PGC1 alpha can also be activated by acetylase enzymes. And then in turn, this activates PPAR, or peroxisome proliferator activator receptor. And this in turn takes an entry into the mitochondria and has ability to act as transcription factors or improve the transcription of mitochondrial genes. So a plenty of enzymes which were regulating this process can actually be activated, and their activity must be stimulated in order to fasten up the process of beta oxidation. So this is one route by which beta oxidation could be controlled. AMPK and PPAR, they play a very important role in this process. Now, second important role, as I told you, is to increase the activity or increase the transport across these fatty acid translocases or CD36. So if you increase the expression or abilities of these molecules, there can be more import of fatty acids, and if there will be more import of fatty acids, then eventually more fatty acid oxidation can take place. But we should know that import is not only enough in cytoplasm, rather, import in mitochondria should also be concurrently increased. So we have a molecule that we looked earlier, carnitine. So if you have increased levels of carnitine that can push these activated fatty acids in large amount into the mitochondria, and they can speed up the process of fatty acid breakdown. So this is the reason that carnitine has also been seen clinically, and there are tablets or injections available which are actually used to promote the beta oxidation or reduction of the fat in individuals to promote the burning of fat. But I strongly advise that this is not a clinical advice to consume this, and you must always consult your doctor before consuming carnitine. This can be harmful as well. So besides increasing the carnitine levels endogenously, beta oxidation is also controlled by another very important link, and that link is fatty acid biosynthesis. So we know that fatty acid biosynthesis and beta oxidation go hand in hand, and they might result in futile cycles if they are being performed at equal rates. But prevention of the futile cycle at one point is prevented by separate compartments. Fatty acid biosynthesis takes place outside the mitochondria, that's in the cytoplasm, and that uses another pool of acetyl coenzyme A, which is cytosolic coenzyme A. So during this process, acetyl coenzyme A is usually converted to malonyl coenzyme A in a rate limiting step, and that reaction is catalyzed by acetyl coenzyme A carboxylase enzyme. Then eventually this goes through a cycle of 4 events, which is almost like a reverse of beta oxidation, eventually adding up one acetyl group through each cycle and elongating the chain. Malonyl coenzyme A dehydrogenase help in that process. Now, since the level of malonyl coenzyme A would be the indicator of increased rate of fatty acid biosynthesis, at that point of time, we would not require beta oxidation. So you can say when malonyl coenzyme A levels are high in the cell, we don't require beta oxidation as such, and therefore it would be inhibiting the transport or movement of fatty acids into mitochondria for beta oxidation. So it will prevent the fatty acids which are being synthesized here from getting exported into the mitochondria and eventually leading to the futile cycles. This is a kind of allosteric inhibitor of carnitine acyltransferase 1. Now, how we control this whole process when we do not require fatty acid biosynthesis but we require beta oxidation to happen? Again, AMP plays a very critical role. AMPK, which is capable of controlling the fatty acid biosynthesis by inhibiting this particular enzyme, acetyl coenzyme A carboxylase. So, if you do not require fatty acid synthesis, AMPK, increased AMPK levels, on one hand, where they're helping in the import of fatty acids, on the other hand, that is also inhibiting this enzyme ACC. And by inhibition of this enzyme, you can expect the decreased level of malonyl coenzyme A. So malonyl coenzyme A would be reduced. And if malonyl coenzyme A would be reduced, in that case, you can expect that carnitine acyltransferase inhibition will be prevented. So you can have again more and more fatty acids entering into the mitochondria, and in that case, there will be increased beta oxidation. So these are some of the critical mechanisms by which beta oxidation is regulated inside the mitochondria, and this is how it goes quickly and much faster whenever required. And whenever fatty acid biosynthesis is needed, at that time, increased malonyl coenzyme A can easily inhibit the beta oxidation process. Let's now talk about something unusual or something different than conventional beta oxidation process. As I told you in the beginning, if you remember, that besides mitochondria, peroxisomes are also the site of beta oxidation. Now, you know the peroxisomes, these are single membrane bound structures and they are mostly known for harboring some of the redox enzymes, and they generate a lot of reactive oxygen species which help in handling pathogens and various other foreign particles or foreign organisms. In case of animals, in case of plants, they may be specialized for carbohydrate metabolism. They are called as glyoxysomes. So these single membrane bound organelles, they have plenty of transporters for different kind of fatty acids, particularly transporters for very long chain fatty acids and normal acyl coenzyme and some of the branch chain fatty acids as well. So if we look at the summary of what common features peroxisomal beta oxidation has and how it is different from mitochondrial oxidation, we would find that first it is doing the oxidation of very long chain fatty acids, which cannot otherwise be done by mitochondria. So these fatty acids longer than 24 carbon atoms, these are actually shortened here. And after shortening, they can be returned back to mitochondria. Now, another peculiar feature which is different and in contrast with mitochondrial oxidation is that there is no electron transport chain here. So reducing equivalents, they don't generate ATP. So irrespective of whatever is the number of carbon atoms in the chain, the net yield of ATP would be zero in this case. However, if you want to correlate that some of the very long chain fatty acids which are broken down here and they got shortened and then they came out and went into the mitochondria, so that can account for some ATPs which are generated, but basically that will be due to mitochondrial oxidation, not due to the peroxisomal oxidation. Now, let us look at the process here, what actually happens. So, all these fatty acids, activated fatty acids, are imported into the peroxisomal cells by these peroxisomal compartments by these transporters. And then they undergo this continuous beta oxidation cycle as usual and generate reducing equivalents. Now, when the chain get reduced to significantly small, like up to carbon number 8, because smaller than carbon number 8 cannot be utilized by enzymes in the peroxisomes. And we should also remember that enzymes here are not identical to the mitochondrial enzymes. We saw their trifunctional enzyme. Here we have a bifunctional enzyme. The structures and functional activity of enzymes is also different in this case. So now these small fatty acids, activated fatty acids, which got smaller here through the successive cycles of beta oxidation, they can now exit the peroxisome and they can eventually enter into mitochondria for further oxidation. Now, besides these very long chain fatty acids, some normal acyl coenzyme can also enter into the routine process of beta oxidation, and some of them can also participate in biosynthetic pathways like synthesis of ether lipids. Now let's look here, branch chain fatty acids. These enter into the peroxisomes, but they have an unusual kind of oxidation mechanism that we will discuss in coming few minutes, called alpha oxidation. So with the help of this alpha oxidation, these branched are actually removed and then they can be further fed into this beta oxidation pathway, again generating reducing equivalents. So now the question is, what happens to these reducing equivalents? If there is no electron transport chain, why does peroxisome carry out this process? So one of the idea behind converting these long chain fatty acids into smaller chain, that's the purpose of peroxisomal beta oxidation. Another, to generate acyl coenzyme for ether lipids. Another, to consume the substrate of alpha oxidation. But these reducing equivalents are actually fed into some of the redox pathways. So you would notice that the NADH, which was generated during this process, can donate its electron to oxygen prevailing in the peroxisomes. And while doing so, it can turn the oxygen into superoxide radicals. Now, these superoxide radicals are quite fast reacting, and they have to be neutralized quickly, otherwise, they will destroy many other biomolecules. So, antioxidant enzyme superoxide dismutase quickly consumes them and turns them into hydrogen peroxide. Now, besides this process, xanthine can also be converted to uric acid in this compartment of the cell by enzyme xanthine oxidase. And during this process as well, superoxides are produced. So these superoxides are again neutralized by SOD, converted into H2O2. Since H2O2 is also toxic, but this can be used to kill the pathogens and other organisms, and this can also be acting as a signaling molecule in the peroxisomes. But the levels of H2O2 can be easily controlled by presence of another enzymes like catalase or peroxyredoxins. So they convert this hydrogen peroxide and utilize this to oxygen. So a delicate balance between all these things keeps this levels of radicals balanced, and signaling is not impaired, and the function of peroxisomes keep on processing because of peroxisomal beta oxidation. Now, besides this intracompartmental production of radicals, there is also a mechanism by which these reducing equivalents can go near the membrane of peroxisomes. And in the membranes, you have some of the enzymes like monodehydroascorbate reductase and cytochrome B, which are electron carriers, and they can take up the electrons and transmit them outside the plasma membrane into the cytoplasm, where they can be taken up by oxygen, making superoxide radicals there as well. So, a lot of superoxide can be attributed to peroxisomal beta oxidation and the reducing equivalents generated there. And these things are implicated in different domains. The redox biology is associated with the progression of cancer, it is associated with aging, it is associated with normal signaling inside the cells. So, we can link this whole process to the redox signaling in one way. Now, defective formation of peroxisomes, if there is a defect, genetic defect which leads to non-formation or defective formation of peroxisome, that leads to some of the disastrous clinical conditions like Zellweger syndrome, which is an autosomal recessive disease. In that case, because it is only organelle which is consuming very long chain fatty acids, if peroxisomes are not there, very long chain fatty acids start accumulating, and that leads to severe neurological problems. Similarly, there is yet another disease called X linked adenoleukodystrophy, where there are defects in the peroxisomal transferase. Particularly, these transferase are defective in this case, and that leads to consequent accumulation of long chain fatty acids, which primarily causes failure of adrenal glands. So these are two clinical conditions which are associated with defective peroxisomal beta oxidation. Now let's talk about yet another type of oxidation that again happens in peroxisomes. If you remember, I talked about the branch chain fatty acids. So one of the branch chain fatty acid that is called as phytanic acid is often found in a lot of dairy products. You can see here that fatty acid is not usual, but it has got several alkyl groups over here. So now the problem originates if there will be substitution on beta carbon atoms. How would enzyme that are digesting or breaking down the beta carbon would recognize it? So such kind of fatty acids which have substitutions or branching, they cannot be metabolized in the mitochondria. And therefore, we need specialized breakdown pathways which are harbored only in peroxisomes. So, this phytanic acid often comes from dairy product and ruminant animal fats and a few fish. It can also be derived from breakdown of phytol groups in the chlorophyll. Now let's look at the process of alpha oxidation. So as you can see here, this first carbon is the carboxyl carbon, but the next one here, this is representing alpha carbon. So primarily oxidation will take place here in this case instead of the beta carbon. In the first reaction, acyl coenzyme A synthetase activates this. As we all know, the first reaction of fatty acid oxidation should be its activation. This is then followed by a reaction that is catalyzed by dioxygenase. Now, this dioxygenase, as you can see here, is introducing an hydroxyl group over here, making this compound as 2-hydroxy phytanoyl coenzyme A. Now, this hydroxy phytanoyl coenzyme A then cleaves formyl coenzyme A from here and leaves leaves a compound called pristanol. Now, this is a ketone which is left over here, pristanol. Sorry, it's an aldehyde which is left here called pristanol, and then this aldehyde is oxidized further by aldehyde dehydrogenase enzyme into pristanic acid. Now, what you saw here, this pristanic acid can easily be then moved one by one. It can be converted further by the sequence of same reactions, same 4 reactions, and this branch chain fatty acids can be broken down. So what we saw here, oxidation was taking place at alpha carbon atom. And what was the product? The product was formyl coenzyme A, 1 carbon compound. So this is an unusual type of oxidation, which is called alpha oxidation, but it does not play a major role. However, if somebody consumes a lot of dairy products and phytanic acid containing products, and that person or individual doesn't have enough enzyme or defective pathway in an individual, that can lead to the severe disorders due to accumulation of the phytanic acid. So particularly a defect in this enzyme, phytanoyl coenzyme A dioxygenase, which is an enzyme that is required for this first oxidation step. Genetic diseases have been linked to it, and one of the disease is known as Refsum disease. So that leads to excessive accumulation of phytanic acid, and that leads to neurological damage. So that was all about alpha oxidation in the peroxisomes. Now finally, let's talk about one of the least commonly occurring type of oxidation called omega oxidation. This is a minor catabolic pathway specially meant for medium chain fatty acids, generally for 10 to 12 carbon atoms. But sometimes it is more important when beta oxidation is defective, and this happens in kidney and liver. So inside the kidney and liver, the organelles which are mainly carrying out this pathway are either microsomes or endoplasmic reticulum. As you can see here in the first reaction, this is the fatty acid which will be oxidized by mixed function oxidase by utilizing molecular oxygen. So you can find here the last carbon atom is actually getting oxidized. And you know that last carbon atom in fatty acid is known as omega carbon. So once that get oxidized, you can see here the CH3 group now became OH. In the next step, dehydrogenase enzyme then takes out the hydrogen from it and further oxidizes, converting this into aldehyde. So fatty omega aldo acid is now formed. And in the next reaction, this is further oxidized by the same enzyme, making it carboxylic acid. So that's a basic logic you may be knowing. Alcohols on oxidation make aldehydes, aldehydes on oxidation make carboxylic acids. So this particular end, or you can say omega end, was oxidized completely into COOH, just like your first carbon atom. So now this has got completely the options. I mean, this whole thing can enter into mitochondria by activation. So normal activation process can happen where coenzyme A can bind to either of the positions, either to first position or to the last position, and then it can go a sequential beta oxidation process as we have seen in the mitochondria. So that's all about omega oxidation. Now, one more add-on thing to be noted here, that these fatty omega hydroxy acids, they are important in the signaling. It has been observed that these hydroxy derivative of fatty acids can serve to produce or inactivate signaling molecules, in particular prostaglandins. So if there is any defect in this particular pathway where hydroxy substituted fatty acids cannot be formed, that is associated with diseases like Crohn's disease and celiac disease. So that was all about beta oxidation and associated type of oxidations. We looked a lot of things, and now I think it's time to just have a quick take home message. So what we learned in this whole lecture was fats and fatty acids are very important energy fuels for living organisms. They help in long haul flights of birds, sustaining waterless life, and our heart, resting muscles functioning. Energetically, fats are more efficient than carbohydrates due to more reduced state and compact nature. We looked at the basic process of beta oxidation that begins from entry of fatty acids into the cytoplasm, its oxidation, then four-step cycle, and then finally generation of reducing equivalents which can make energy. So fatty acids are mostly oxidized in mitochondria via sequential oxidation of beta carbon, and thus it's named as beta oxidation. And finally, we emphasized on calculation of ATP. So we have looked at development of a formula for most commonly occurring fatty acids that are even numbered fatty acids. The ATP yield can be given by 7C minus 6, whereas for the odd numbered it can be given by 7C minus 19. We have also looked at other formula for unsaturated fatty acids as well. And then other routes of oxidations also exist. One, peroxisomal beta oxidation, peroxisomal alpha oxidation, and from the other side of the carbon, that's omega carbon or omega oxidation. So that's all from my side. I believe that you enjoyed learning beta oxidation. Thank you very much for your patience and best wishes.